My name is Dawood Darbar, and I'm a clinician scientist, cardiologist, and cardiac electrophysiologist. I'm also Chief of Cardiology, Professor of Medicine, and Associate Program Director of the Medical Scientist Program at the University of Illinois at Chicago. I first became interested in biomedical research as a medical student at the University of Dundee, Scotland, when I pursued an intercalated Bachelor of Medical Sciences degree. While my research was focused on the evaluation and management of patients with hyperlipidemia, I am most passionate about the translation of cardiovascular pathophysiology from bench to bedside with a focus on cardiac arrhythmias, a major public health problem. This interest has been enduring and led me to pursue a doctorate at Vanderbilt University, known for its strength in basic and clinical and translational research within the Division of Clinical Pharmacology, and their commitment to the development and mentoring of young clinician scientists. The goal of my research since its inception has been the translation of cardiovascular pathophysiology from bench to bedside with a focus on cardiac arrhythmias, especially atrial fibrillation. Atrial fibrillation affects 2 to 5 million Americans and continues to be a major cause of morbidity and mortality. Until recently, atrial fibrillation was considered to be a sporadic, non-genetic disorder, but studies at UIC and other centers have identified both common and rare genetic variants contributing to the risk of developing atrial fibrillation. Although the underlying mechanisms have not been completely worked out, our work and that of other centers clearly suggests that atrial fibrillation is a genetic disease with variable age-dependent penetrance determined by diverse genetic and acquired arrhythmogenic mechanisms. I have developed a research program that addresses both clinical management as well as the underlying genetic issues in atrial fibrillation. I established the Vanderbilt Atrial Fibrillation Registry, a key enabling resource for our studies that now includes over 3,000 subjects and their clinical data as well as DNA samples. More recently, we established the UIC Atrial Fibrillation Registry, which has helped us to identify not just the clinical and genetic and molecular subtypes of atrial fibrillation, but also lay the groundwork for long-term vision of replacing empiric treatment for atrial fibrillation with mechanism-based therapy. The biorepository's major focus on ascertainment of families has allowed us to identify novel genes, not just in Caucasians, but also in African Americans, and more recently in Hispanic Latinos with familial atrial fibrillation. Using traditional linkage analysis, candidate gene, and next-generation sequencing, we have elucidated the underlying molecular and genetic mechanisms of atrial fibrillation, but also how patients respond to different treatments based on their genetic makeup. My lab's ability to functionally characterize novel mutations and genes using in vitro and in vivo models, and more recently ex vivo induced induced pluripotent stem cell-derived atrial-like cardiomyocyte has allowed us to uncover the underlying genetic and molecular mechanisms of atrial fibrillation. The clinical DNA biorepositories have also been a key contributor to national and international genome-wide association studies that have also afforded us the opportunity to build collaborations with an international consortium of investigators. My research interests marry perfectly with my clinical practice as a cardiac electrophysiologist, where I look after patients and their families with inherited arrhythmia syndromes, including atrial fibrillation, and provide individualized care. So in summary, at UIC, we have developed an outstanding and highly successful clinical and translational program focused on delivering personalized therapy that spans the whole spectrum from molecular, cellular, and animal studies to human translational trials testing novel hypotheses and defining the underlying pathophysiology of inherited arrhythmias. I worked in the laboratory of Dr. Dan Roden, one of the world's leading experts in the mechanisms and genetic determinants of cardiac arrhythmias and response to drug therapy. He is an outstanding mentor who has trained over 100 students in his 30-year career and is particularly effective at identifying mentees' strengths and weaknesses such that they can build on the former and correct the latter. Through his example of boundless enthusiasm and delight at scientific discovery, Dr. Roden instilled in me a deep respect and thirst for new knowledge. My goal is to continue this tradition of outstanding mentorship with the training of the next generation of clinician and physician scientists, and to instill in them a passion for scientific discovery that will be sustained throughout their own academic career. I truly believe that clinician and physician scientists are uniquely qualified to have the greatest impact on the care of patients. It is also a privilege that we must not squander and make the most of. The research questions that my lab tries to answer come directly from taking care of patients. For example, the most common treatment for atrial fibrillation is only effective in about half the people. Why is that? I truly believe that a physician scientist or clinician scientist is best able to answer that question. We recently showed that in addition to the genetic makeup of a patient, other factors such as obesity seem to impact how an individual patient responds to treatment for atrial fibrillation.